I will be talking about integrative analysis of single-cell omics and imaging data. So I was gonna just mention this also, since Andrew already mentioned. This presentation is part of a four-part webinar series focusing on spatial multi-omics. I'd like to take this opportunity to thank the folks at Akoya for organizing and sponsoring this very exciting series, especially Carla Mulit and Oliver Braubach and Nilima Kumar. So I will start my presentation with some background on current single-cell omics and imaging technologies and challenges in data integration. Then I will describe a computational tool we recently developed for integrative analysis of single-cell omics and imaging data. So as you know, there has been a long interest in understanding tissue microenvironment. In recent years, this is further stimulated by the advance in single-cell technologies. This webinar series is actually a very excellent example of this development. So my lab is particularly interested in understanding tumor microenvironment. As you can see in this cartoon, the microenvironment is really complex. This cartoon highlights the various types of cell-cell interactions between the malignant cell on the bottom and stromal cell on the top, interactions among the stromal cell themselves. Now, admittedly, this cartoon is based on knowledge learned mostly using bulk analysis. So it's probably there's fair amount of underestimate of the role of heterogeneity in the tumor microenvironment function. Speaking of heterogeneity, it's manifested at all levels during the information flow from DNA to protein. So we have genetic heterogeneity, epigenetic heterogeneity, transcriptomic heterogeneity, and proteomic heterogeneity. All these we are getting better handle to understand using single-cell technology. Finally, there's an increased appreciation of the microenvironmental heterogeneity. In other words, the spatial heterogeneity of the tissue, how it plays a role in the tissue function. In the recent years, there have been tremendous development in single-cell technologies, which we can break down to omics-based technologies and imaging-based technologies. Although this division is getting blurred because of the advent of in situ sequencing technologies. Nevertheless, for single-cell omics assays, we now have high throughput technologies that can measure a number of modalities within the cell. For instance, we have a technology to measure histone modification at single-cell level, as well as DNA modification. We can also measure the chromatin accessibility, as well as three-dimensional chromosomal organization at single-cell level. Finally, we also have the well-known technology, single-cell RNA-seq, to measure the transcriptome. For imaging-based assays, we have high-throughput assays for measuring both protein and RNA. For protein, codex is a powerful representative technology. It has several very attractive features shown here. First, it has a very high plex in terms of number of protein it can interrogate. Typically, we can measure more than 40 proteins in a single experiment at single-cell resolution. And secondly, it can image the entire tissue section instead of selected fields of view. And finally, it is very easy to use with any one staining step followed by automated imaging session. This is one example codex image. So codex protocol can use both formalin fixed tissue section as well as fresh tissue section. In this particular example, it's FFPE fixed sample for human breast cancer. As you can see, this is the entire tissue section imaged. If we zoom in, this particular antibody panel consists of 36 antibodies targeting various epithelial cell types, as well as different cell states and proliferation markers. In this particular zooming example, we are looking at four colors, including the nuclear DAPI staining, the lamine B1 stain nuclear envelope. E2F1 is malignant cell transcription factor, and E-catenarin that stain for the epithelial cell. You can see very high resolution of individual cells and even subcellular resolution staining the nuclear and nuclear membrane. This is another example using fresh frozen human rash sample. Again, you can see this is a very high resolution image. We can clearly see the basal cell membrane stained green using CD49F, as well as inflammatory leukocytes stained using the pan-leukocyte marker CD45 in the dermis. So beyond these two examples, Akoya has tested the technology in a range of human and mouse tissues, as shown here, using both formalin fixed tissue section as well as fresh frozen tissue samples. So another category of multiplex imaging protocol is the very high multiplex single molecule RNA FISH protocols. There are several of those protocols. Shown here is a representative assay type called multiplex error robust FISH or MER-FISH. It is developed by Xiaowei Zhuang's group a few years back. In this cartoon, you can see MER-FISH can simultaneously interrogate several hundred to over 1,000 RNA species in the tissue section. On the right, you can see the measured RNA expression level is highly concordant with the RNA expression level measured using single-cell RNA-seq, as in panel D. So both the MER-FISH and codex, therefore, represent a very powerful spatial imaging platform to understand tissue microenvironment. So all these single-cell assays, both omics-based and imaging-based assays, they only interrogate one aspect of the cell state or the microenvironment state, as shown in this cartoon. As we all know, the cell state is represented by all the different molecules inside the cell. So ultimately, what we want to do is to integrate the different modalities to get an overall sense of the cell state or the microenvironment state, how these play a role in development and disease. Hence, the focus of this part of the webinar series. Multiomics data integration is clearly very challenging. This is not a brand new topic. This has been researched for quite a while. In the past, you're focusing on bulk omics and imaging data. So there are many challenges. Here are some of those I came up with. In no way a complete list. Some of those challenges are common for both bulk and single-cell data, but some of those are unique to the nature of single-cell data. Let me go through these challenges. First of all, a typical issue with imaging data is they tend to have much fewer number of features compared to omics data. For instance, you typically only have hundreds of smaller than 100 features for imaging data. Compare that to thousands of features for the single-cell RNA-seq data, or even tens of thousands of features for single-cell epigenomic data. So that creates a challenge. Secondly, in order to integrate the different modality, the first step is to identify a common feature space, so the additional calculation can be done on this common feature space. So it is still open question, what is the best way to identify this common feature space? There are different approaches proposed, but in my opinion, it's still a fairly challenging question. Part of it is due to the biological nature of the different molecules that are captured. For instance, the RNA is quite different from epigenome. How do we first process this data so we can start to compare them? Is still an open question. And thirdly, this is a unique feature of single-cell omics data, which is that they are inherently sparse compared to bulk data. And the omics data is even more sparse, meaning there are more dropout compared to the imaging data. How do we address this is also need to be considered. Finally, there is generally a lack of standards and data set for evaluating the performance of these integration methods. I think this is the issue that we as a community should start to address together. So with all these challenges, there are already a few methods that in the recent year have been developed for data integration. Two representative methods are shown here. The first one is a SORAT. The other one is a Liger. These are very powerful methods for single-cell omics data integration, but in my opinion, there's still a large room for improvement for this very important topic. To this end, my lab also recently worked on this problem, and we developed a computational algorithm we call GLUR for integrative analysis for single-cell omics and imaging data. This is spearheaded by a very talented postdoc, Tao Peng, in my lab. So for the next few slides, I'd like to briefly go through the major algorithmic steps of this method before I show some performance analysis using omics data. So in GLUR, the different data modalities are all represented as a matrix. The rows of matrix is the data modality specific features. For instance, transcriptome data will be RNA-seq, will be transcripts. Epigenomic data could be genomic region or interactions or DNA methylation level, depending on data modality. And for imaging data, if you're using codex, the row will be proteins and so on. And the column are cells. With these matrices as the input, GLUR has three major algorithmic steps. The first step is finding that common feature space, as I mentioned. To do this, we use a mathematical framework called joint non-negative matrix factorization. So this framework has a very attractive feature because, as the name suggested, it deals with non-negative values. So the optimization is constrained by constrained to non-negative values. So this is very natural because all the data types we are dealing with are non-negative values compared to principal component analysis. This is more natural to interpret the result. So what comes after this, come out of this joint non-negative matrix factorization is a set of low-dimensional features that are shared by the different data modalities, or we call it the latent feature space. And then in step 2 of the algorithm, we try to match the different cells from the different modality using algorithm called mutual k-nearest neighbor. So this is done in the shared feature space instead of the original features. One of the data modality we treat it as a reference modality. The other modality we treat it as a query modality. The cells are matched based on the similarity of their mutual neighborhood in the latent feature space. And then in step 3, with the matched cells, we like to calculate mapping function between the latent features from the reference modality to the latent feature of the query modality. So we do so using artificial neural network, as shown here in this cartoon. So for each pair of query and reference modality, we train one neural network model, as shown here. And if you have 3 modalities, then you will have 3 neural networks models. So what comes out of this is the model. A neural network is a mapping function that can map the reference latent feature value to the query latent feature values. With this mapping function, we then can co-embed or integrate the different data modalities. And then with the integrated data, we can do a number of things. First of all, we can certainly do imputation both for the omic data and the spatial imaging data. And then those can be used for data type identification. And furthermore, we can use those data to try to understand the transcriptional regulation and the signaling pathways between the different cell types and so on. So as I discussed before, the best way to evaluate the performance of those integration methods remain an open question. So here's a strategy we adopted in this project. So in order to evaluate the performance, we need 2 things. One, first is we need to know the ground truth of cell-cell pairing from the different modality. To address this, we take advantage of some public true multi-omics data sets. In particular, we used dual omics data that can measure both RNA and chromatin accessibility, or ATAC-seq, in the same individual cell. I will explain how we use this type of data in the next few slides. And then the second issue is what is the performance matrix we want to use. For this, we consider 3 types of matrix. The first 2 are semi-quantitative, and the third one is more quantitative and more global. The first criteria we use is what is the degree of mixing of cells of different modality in the integrated data. And secondly, what is the degree of separation of cell type or cell states in the integrated data, if we know for sure what are the cell types in the input data. And finally, we devised this integration accuracy metrics to globally measure as the name suggests, the accuracy. I will explain these in more detail in the next few slides. Okay, so we in this project we tested the performance of Gluer using a variety of publicly available single-cell omics and imaging data. In this first exercise or analysis, we use this dual omics data type called SNARE-seq, developed by Kun Zhang's group. So a simultaneous measure RNA and chromatin accessibility in the same single cell. In this particular data set we used, the sample is a mixture of 4 different human cell lines, BJ, H1, K562, and GM12878. So in this case, we know for each cell we know exactly what is its type. So let's look at the performance of the 3 algorithms, Seurat, Liger, and Gluer, in terms of the 3 metrics I just introduced. First of all, let's look at the mixing of the different modality in the integrated data. Shown here are UMAP, basically a dimensionality reduced plot based on the integrated data. The blue dots represent cells based on the transcriptome data, and the orange dot represent cells from the chromatin accessibility or ATAC-seq data. The first thing you'll notice actually of the 3 methods, the Liger integration result appear to not mixed well as the other 2 methods, Seurat and Gluer. And secondly, if we look at the cell cluster, you can see Gluer integration result yield a very nice separation of the 4 different cell types that are color coded. So we can confirm the cluster identity using known cell type specific marker genes, as shown here in this violin plot. Again, you can see compared to Seurat and Gluer, the marker gene expression is not as specific as the other 2 methods. So this is consistent with what we observe in the UMAP, global UMAP. And finally, we devised this global metric called integration accuracy. It is defined as the percent of true cell pairs among the predicted neighbors of a given cell. In this case, let me explain how we do that. In this case, because we use the dual omics data, we basically, when we run the different algorithm, we basically separate the RNA from the ATAC-seq data, pretend they are from different, they are measured separately. Then we perform the integration. And when it comes to integration accuracy, we know for each cell what is this true cell pair from a different modality. Therefore, we can calculate the percentage true cell pairs. As you can see here, using this metric, Gluer has significantly higher integration accuracy globally. And then we evaluate the performance of Gluer using primary tissue samples. Here again, we use SNARE-seq data set on mouse cerebral cortex data. In this particular analysis, Seurat doesn't the integration result of SORA doesn't mix the modality very well, as you can see here. The LIGO result mix the modality pretty well, but it's hard to see the cell cluster structure in the UMAP. On the other hand, GLUR has both well-mixed cells and good separation of cell clusters. These clusters can be identified using known cell type marker gene. This is shown in this slide. As you can see, each row is a different cell type. Each column here is a particular cell type, known cell type marker gene. The size of each dot represents the expression specificity of a marker gene in that particular cell type. So as you can see, the GLUR based integration result generate clusters with that has a consistent gene expression pattern using those marker gene. And furthermore, on the bottom is the ATAC-seq signal profile across these different clusters, which is concordant with the expression profile using RNA-seq data. And finally, we can see the global integration accuracy of GLUR is significantly higher compared to the other two methods. So that is the performance evaluation for GLUR. For the rest of my talk, I will present how GLUR can be used to integrate single-cell omics and multiplex imaging data. And first of all, I'd like to show the utility of integrating single-cell RNA-seq data with codex data. So in this particular example, we used a codex data set on mouse spleen published by the Nolan lab. In this particular data set, they used a 30 antibody panel, which covers the majority of spleen cell type. This is the matched single-cell RNA-seq data of mouse spleen generated by Pablo Camara's group, who is the next speaker of this webinar series. Again, you can see it captures the majority of the spleen cell type shown here. So let's look at the integration result. The before and after integration using GLUR. Shown here on the left is the original data before integration. Clearly, you can see the RNA-seq data, as expected, already shows some very nice cluster structure in the UMAP, whereas for the codex data, the cluster structure is less pronounced. This is as expected, as I mentioned, because there are much fewer features in the data, but there are more cells captured using the imaging modality. And then this is the integration result using GLUR. On the top is RNA-seq data. On the bottom is the codex data. You can see very clearly there are many clusters even in the codex data. And the two data modalities, the well mixed on the upper right corner, you can see that's the co-embedded data. But noticeably, there are certain clusters that are mostly driven by the codex data, suggesting that codex data may capture some subtle cell state that not captured by the RNA-seq data. On the bottom right corner, you can see the different cell types annotated using known cell type marker gene. So the integrated data clearly show cell type specific expression both for RNA and protein. This is one example. So the VCAM1 is a marker gene for red pulp macrophage. It is highly specific to cluster number 12, both based on the RNA-seq data and the codex data. Note that although it is a known marker gene, but it is hard to use this marker gene to identify that red pulp macrophage cluster based on the codex data alone. Hence, emphasizing the importance for data integration. So this is another example. So the NCR1 is a marker gene for NK cell. In this case, it's highly specific to cluster number 16, which is annotated as NK cell. Again, you can see both the RNA and the protein level is highly specific to this cluster. And for the rest of my talk, I'd like to show you how we can use Gluer to integrate RNA-seq data with another multiplex imaging modality, which is the multiplex single molecule RNA-FISH protocols. In this particular example, we used a variant of protocol from Merfish. It's called SigFISH, for the reason that there is a matched single-cell RNA-seq data. For this SigFISH dataset, they used the mouse olfactory bulb tissue. As you can see here, the dataset capture a variety of cell types in the tissue, including neuron, different glia cells, and endothelium cell, mitral cell, etc. This is the single-cell RNA-seq data. From a different group. This is the before and after Gluer data integration. Similar to the codex analysis, again, you can see in the original data, there's a very pronounced structure in the single-cell RNA-seq data, whereas it's less so in the SigFISH data. But after integration, first of all, you can see the data modalities are well mixed, and more importantly, we see a number of clusters representing different cell type, and the cell type from different modalities are also clustered together. In this legend, cell type name with the S as the postfix represents SigFISH data. Cell type with the postfix NS represent single-cell RNA-seq data. You can see for many cell types, the RNA-FISH data is right next to the single-cell RNA-seq data. Apologize for this little font size, but you can see many of them are next to each other. Suggesting the integration is successful. And with that, I like to conclude. So I present a new computational algorithm called Gluer. We designed it as a general framework for integrating single-cell omics and imaging data. In terms of the algorithm, it combines three computational framework: joint non-negative matrix factorization, mutual k-nearest neighbor, and artificial neural network. And we evaluate the performance of Gluer using a diverse set of data and show improved integration accuracy by Gluer. With that, I'd like to thank my lab members who spearheaded this project, mostly led by postdoc Tao Pen, with help from Greg Chen, a graduate student, and Yu Xuan Hu, another graduate student. And I'd like to acknowledge our collaborators and funding resource. Thank you for your interest. I will stop there, take questions.
Thank you, Kai. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. Please remember to take a moment to fill out our survey questions now by clicking on the green survey icon at the bottom of your screen. We will now start the Q&A portion of the webinar. And the first question is Gluer publicly available?
Yes, it is deposited in GitHub. And the manuscript describing the algorithm is on bioRxiv as well. There is a link to the GitHub depository.
And is there a graphical user interface available for Gluer?
Yeah, in the software package has a graphic user interface, but it's mostly at the current version only implement functionalities that help you explore the result of the integration. But we are working on the first part of actually running the algorithm using graphic user interface. So we will update it soon.
Is there a particular computing setup that you recommend for running Gluer?
Good question. Gluer is not computationally demanding in terms of the resource. We even run it on a personal computer. So yeah, I would say it should be usable on personal computer as well as high-performing environment.
Why did you decide to use UMAP for dimensionality reduction over another method such as Trimap?
Oh, good question. What is Trimap? I'm not familiar with Trimap.
I wasn't either. It's another method of dimensionality reduction using something called triplets.
Triplets. Okay, yeah, I'm not familiar with that. You know, the other commonly used one is the t-SNE, but there are studies showing UMAP is generally a better algorithm than t-SNE. But sorry, I'm not familiar with Trimap.
Do you have drop-off of cells, and to what extent might there be that drop-off?
Yeah, great question. Certainly, for sure, there's dropout in the omics data set. Because we use the public data, I would refer you to the original publication of those data sets. But I'm 100% sure there will be dropout. It's generally believed that it's less so in the imaging data.
And then would it be possible to combine Gluer with with some dropout fixing algorithms for single cell RNA sequencing like MAGIC?
Yeah, that's certainly a great suggestion. That can certainly be done at least as a pre-processing step. But if we want to include it as a in a principal way into the algorithm, yeah, that will require some additional research, but that's certainly a very interesting direction.
How did Seurat and Liger integration methods compare with Gluer when integrating codex and single-cell RNA-seq datasets?
Yeah, I didn't show the result here, but in general. So this has to do, so as I mentioned, it's still an open question, how do you evaluate the performance? In the case of a single cell omics integration, for instance, RNA-seq and the ATAC-seq data, we know the ground truth based on the dual omics data set I mentioned. But in the case of integrating omics with imaging data, to the best of my knowledge, there is no gold standard data set. So, but we did look at just visually, we see there's more, as I mentioned, using these two semi-quantitative measurement, looking at the separation of the cluster, we see more separation of the known cell types in the data set. Using that as a metric, then I would say there's also improvement there.
And do you think that your method would work with consecutive slides, one made with codex and the next one made with 10x Visium?
Yeah, that's a great question. To my knowledge, the Visium technology right now is not at a single cell resolution yet. So that will create some challenge how you represent the data as input, because the codex, each row, well, each column is a single cell, but the Visium right now, you will have each column is a meta cell, so-called meta cell, representing a number of several cells. So that's a difference. Certainly, you can try to impute the Visium data into single cell, and then do integration that way. But that's another layer of manipulation. Or you do another way, try to combine the single cell from codex into a meta cell to integrate. So yeah, so there's something we need to consider, what is the best way to do that. That's a good question.
Next question is, do the different data sets to integrate have to measure the same cell population? And what if one data set measured a small subset of the cell population as another data set?
Yeah, so I'm assuming you are asking maybe the same data modality, but different measuring different cell population. So the gluer, if that's the case, gluer is, as I mentioned, it's a general framework. So then you can treat the different experiment as an individual matrix, and then you can integrate that way, if I understand the question correctly.
What is the process for annotating cell types once the merged cluster map is formed?
Yeah, that's another great question. In the examples I showed, we mostly use known cell marker genes. So it's mostly driven from RNA-seq side, or we can, if for codex, we can also use certain known cell type specific protein markers. Because of the data are integrated, we can annotate the epigenome data from the RNA gene expression side. But we do see for certain clusters, the correlation between the gene expression and the epigenetic feature is not very high. So this is the question I alluded to in the introduction. How do we capture the different features from the different modality as input for integration? Seems to me it is an open question. What's typically done, let's say take ATAC-seq data as an example, what's typically done is they summarize the ATAC-seq sequencing reads in the promoter and gene body of a gene. Therefore, you can match, so the common unit is the gene for both RNA and ATAC. But clearly, we know for gene expression, there are many distal DNA element that have very strong signals for ataxic data. So these kind of information is not used yet in many of those existing methods.
Next question is, how do you take into account the spatial relationships between different cells that can be detected with codex?
Yeah, in Gluer, we don't use this type of information because we have to figure out, if we want to use this kind of information, we have to figure out how we represent that information as input for integration. So what we can do, where I haven't shown yet, is after integration, we can certainly go back to the original codex or imaging data to try to identify certain spatial patterns, for instance.
All right. How does one determine the marker genes for any particular cell?
Yeah, that's a great question. In our this project, we mostly just based on existing literature evidence, but certainly identifying cell type or cell state is also a open question for the single cell field. There are no algorithms for automated cell type annotation, but in this project, we want to make sure what we use is robust. Therefore, we resort to the existing literature.
Have you tried using Gluer to integrate more than 2 data modalities?
Yeah, that's a great question. We haven't. The reason for that is because if for discovery point of view, yeah, we can do that. But for evaluating the performance, the problem there is the identifying a gold standard data set that have 3 modality measured in the same single cell. To our knowledge, identifying this kind of data set, add separately the different 3 modality separately, we haven't looked at that. To my impression, I'm not sure such a data set exists, but if people knows about that, please let me know.
Is there a minimum number of codex features required for good integration with single-cell RNA sequencing data?
Yeah, that's an interesting question. We haven't tried to downsample public codex data set. Yeah, that will be something interesting to do. But again, the question is how do you evaluate the performance? Right, so that's something we need to think about it.
Do you recommend a method to map enhancer regions from epigenomic data to genes?
Yeah, so that is definitely one way to go. We have tried that, but so far it's not very successful yet. I think the reason for that is mapping enhancer to target gene itself is a very challenging question. If the accuracy is not very high, especially at single cell level, then that might actually introduce more noise than signal. But certainly that's something I think is a direction to go, but I'm sure probably people are working on that, but I haven't seen really very exciting breakthrough yet.
Do you recommend restricting the clustering when analyzing codex and RNA-seq data?
Restricting clustering. Well, first of all, let me explain. So the integration doesn't require clustering. After the different algorithm integrate the data, then we take the integrated data or the co-embedded data to do a clustering using the same clustering algorithm. In this case, I think it's a Louvain clustering, the community detection algorithm. Hopefully that answers your question.
Does GLUER provide corrected gene expression values that could be used for additional downstream analysis other than dimensionality reduction and clustering, such as differential gene expression and pathway enrichment analysis?
Right. It provides the the co-embedded values for all the modality integrated. So you can certainly do a downstream analysis, as I mentioned in one of the slides.
Is there a danger of overfitting codex data when using GLUER? Does the GLUER algorithm need to be adjusted or fine-tuned from one data set to another?
Yeah, that's a great question. We haven't addressed it, but certainly that's a possibility. This also has to do with the setup of which data modality you use as a reference versus a query. So this, I can imagine this can be addressed several ways. You can change the definition of a query versus a reference. You can also downsample a data modality versus other. Yeah, that's definitely a great suggestion.
Sorry about that. Is the GLUER output an ANN data object that allows for downstream analysis in Python?
Yeah, it is. It's actually implemented in both. The bulk of it is using Python and some R scripts. Yeah, it is.
One audience member has a question. By combining single-cell data with imaging, aren't we compromising on the spatial resolution? How do we co-register data with different inherent spatial resolutions?
That's interesting question. I guess this has, if I understand the question correctly, this one has something to do with the previous question, imaging data with different resolution. But I would think ideally you still want to go down to single cell resolution because the other modality are single cell resolution. In that sense, I don't know if you will lose information. You might have more, well, I guess it's hard to say. The cell segmentation has some noise, but that's the how do you deal with the image analysis, not necessarily a problem for integration per se.
Okay. Can you use GLUER to find the key imaging features related to disease progression or drug efficacy?
Yeah, that's the ultimate goal, right? So, but GLUER just provides you with initial integrated data, and then from there, there need to be some downstream analysis. So, for instance, you find spatial patterns that has both transcriptome, epigenome information attached to it, and then you have to compare different conditions in order to draw a conclusion. For instance, for a disease study. So, yeah, in itself, Gluer just provides the first step, I would think, but a critical first step.
From your experience, do formalin, fresh frozen, or Perelman embedded tissues provide different data retrieval?
Data retrieval. This is an experimental question. I don't know what that means by data retrieval. Are you talking about the antigen retrieval? Because FFP definitely you need to do antigen retrieval.
I think if I had to rephrase the question, it would be: do different tissue types provide better data quality for use in Gluer, and from the codex side of the input?
Yeah, I would imagine probably true, but that's that itself it's not necessarily a question for the integration part. It's a question for the codex technology part. But I guess, sorry, maybe just add one more thing. Maybe, yeah, in the future development, we can assign some kind of confidence measurement to the input data that can be considered in the integration step. That maybe will address this question.
All right, and then we have a question about multi-omics data. Can we use Gluer for true single-cell multi-omics data, genomics, proteomics, epigenomics, and metabolomics, et cetera?
Yeah, that's so you would think the true omics data doesn't need to. I mean, the integration is straightforward, right? But actually, what we found is actually we just tried Gluer on true omics data integration. For instance, ATAC-seq plus RNA-seq. In there, we basically we don't need to deal with the matching cell label from different modality, but we apply the three methods. We found actually we still get quite a significant improvement in terms of identifying the different clusters and the known gene cell type specific gene expression. So I would argue even for true multi-omics data, the integration procedure is not necessarily straightforward.
All right. Well, that's all the time we have for today. We'd like to thank Kai Tan and our sponsor, Akoya Biosciences.